Recording. Awesome. All right. Great. And our projectors are working, so it's already a good day. My voice is probably the worst it's been because I was at the track and field worlds last night. So I was screaming for track athletes and excitement. So I will keep it together. But I apologize if I'm a little more scratchy than usual. We swept the 200 for men, and then it was exciting. It was just a good day for track and field. All right. Oh yeah, awesome. I appreciate that. Awesome. All right. So reproductive system, we're going to get into some general pieces of the reproductive system that are, I guess you could say, the same for male and female gendered. And then we're going to move into male gender anatomy and some physiology. So a reminder here, when we start at the beginning, we have the same 4 tasks that start in the reproductive system, whether you're male gendered or female gendered. So both are going to form specialized cells for sexual reproduction called gametes. It's important that we know these terms. They love to throw out these terms, gametes, ova, zygote, all these things. So keeping them separate in your head will be important. So again, we have gametes. Sperm is the gametes in males. Ova is or eggs is the gametes in females. Gametes for males and females come together through sexual intercourse or copulation. That's 2 terms they'll use. We combine genetic information then through fertilization, and that's when we get our zygote, is when our egg and our sperm fuse. The zygote is that first cell of new individual, and all body cells arise from it. And we'll talk a little bit about cell division and how that process occurs, just as a reminder for y'all. When we think though about our accessory reproductive organs, they're homologous structures. What does that mean? They share common origin during development. So the male testes and the female ovaries share common origins. The male penis and the female clitoris share common origins. So primary sex organs that we can consider would be our gonads, our testes, and our ovaries. And they have 2 specific products, our gametes, which we've talked about, which are going to form by cell division via meiosis. We'll do a quick rundown of meiosis. We won't spend a lot of time on it. And then our sex hormones, our steroid hormones, so testosterone in males and estrogen and progesterone in females. So we are going to have a little bit of a reminder of some of our HPA axis and our adrenal hormones and our endocrine hormones, because we're going to talk about the HPG axis a little bit today. We also have accessory reproductive organs, which are ducts, glands, and external genitalia, which we'll touch base on development. So our HPG axis is our hypothalamic pituitary gonadal axis. And this is where we're thinking about what was produced in the hypothalamus, the pituitary, and then what effects does it have on those primary organs. So our hormones involved is GnRH, FSH, LH, testosterone, and inhibin. We'll talk a little bit about some of those hormones, but we'll skip the ones we've already covered. So we've talked a little bit already about gonadotropin releasing hormone, GnRH. It's released from our hypothalamus. It's going to reach our anterior pituitary, which then is going to stimulate the release of FSH and LH, which we've talked about before. So then downstream, once FSH and LH are released, that's going to stimulate testosterone, estrogen, and progesterone formation and release at specific target tissues in our body. And then inhibin, this is fantastic. It is going to inhibit some of these hormones from being released. It's one of the easiest ones to remember, right? Inhibin sounds like inhibit, great. But it does that through negative feedback control on FSH release. So you can think about inhibin is going to inhibit FSH release from your anterior pituitary. And what does that control pattern look like? Looks like this. We've seen this before. Our hypothalamus, anterior pituitary, gonads. Now at puberty, we haven't talked about what happens, the changes that happen then at puberty and what that looks like on a cellular and hormonal level. So at puberty, the HPG axis is activated, right? This is the period of life where our reproductive organs have grown to our adult size. They can continue to grow and develop, obviously, as we know, but they become functional. And this is the earliest time that reproduction through fertilization is possible. Before puberty, low levels of circulating steroid hormones are enough to kind of suppress that GnRH process, that GnRH release. But as puberty approaches, the hypothalamus becomes less sensitive to inhibition by sex hormones. And you could almost think about it as like a pulse that gets stronger and stronger from the hypothalamus with that GnRH or GnRH release. So once the pulse of GnRH gets high enough, right, once we reach that threshold in the body, then the anterior pituitary is stimulated enough to start releasing FSH and LH. And then that stimulates the gonads to release more sex hormone. And then that's how we start getting our effects from puberty, whether it's a menstruation cycle for females or secondary sex characteristics in both male and females. And we'll look at a few of those pieces when we get to those specific gendered organs. But here's a quick memory lane. We're going to walk down memory lane of meiosis, just because there was some questions about meiosis in the past. So if you remember, meiosis is not mitosis. Mitosis is general cell replication, right, and division. Meiosis is unique and that it only occurs in your gonads. It's the same process for males and females, and sexual reproduction can't take place without it. You're going to reduce the number of chromosomes in your gametes by half, so your zygote doesn't end up with double the chromosomes. And this is where a lot of our genetic congenital conditions could start with, especially if there's abnormal chromosome numbers or genetic defects. So understanding kind of the generalities of meiosis is helpful in being able to walk yourself through what is happening in some of these congenital conditions. So first, just some terminology, so we're all on the same page here. Our sister chromatids, so cells' DNA is replicated during interphase. Our sister chromatids is each chromosome has two of them, two sister chromatids. They're identical. They're joined together by a centromere. I teach genetics, so let me know if I'm going too slow or too fast, because I'll just speed through this otherwise. So each chromosome, two sister chromatids joined together by a centromere. Then we have DNA that's going to coil and condense, making our chromosomes visible, because we can't see them normally until it condenses out. And then our homologous pairs of chromosomes, one member of each pair is from the father, paternal chromosome, and one is the maternal chromosome. Okay? They look alike, they carry the same genes and code for the same traits. Then we have our non-sister chromatids. Within our homologous pairs, the chromatids of one chromosome are non-sister chromatids to the other chromosome in the pair. The non-sister chromatids are the ones that get to do the DNA exchange. They do this cross-linking pattern. They cross over. And this is how we have increased genetic variability. That's why we don't all look identical, right? When we're thinking about numbers, there's two different numbers that can tell us how many chromosomes are present, and they're diploid and haploid. So diploid chromosomal number is the normal number of chromosomes in most body cells. 2n consists of two sets of chromosomes, one from each parent. 2n is how it's represented. Our diploid number, therefore, is 46, which means we have 23 pairs of homologous chromosomes. Okay, so our normal diploid number is 46. That means our normal chromosomal pairs is 23, half of 46. And that is normal. So anything that's not 46 or 23, right? Diploid 46, haploid 23 is abnormal. Our haploid chromosome number is N, which is the number of chromosomes in a gamete. Each gamete contains only one member of each homologous pair, which is why when both gametes come together to form a zygote, we don't have double the amount of genetic material. So our haploid number is 23. That means when sperm and egg fuse, they form a diploid, 2n, 46 chromosomes or 23 pairs, which is a zygote. So our meiosis phases, again, we're gonna fly through this, is meiosis 1 and meiosis 2. DNA replication in this process occurs only one time. The product is we get 4 haploid, so N, 23 chromosome daughter cells. And each nuclear division, so meiosis 1 and meiosis 2, involves the prophase, metaphase, anaphase, and telophase that we know from mitosis. So this picture, I think, is a little bit easier just to visualize if you're a visual person like me, especially right in the morning, it's hard to track numbers. So we're going to follow one pair of homologous chromosomes through our interphase and meiosis process. We have our interphase here. We have our pair of homologous chromosomes in our diploid parent cell, 2N, 46 total, right? The chromosomes are going to replicate. So we have double the fun, double the genetic material. These are their pair of replicated homologous chromosomes. Meiosis then begins with a diploid parent cell that has replicated chromosomes in the middle. In meiosis 1, the homologous chromosomes are going to separate. So we still are going to have that replication of those two chromosomes. They're separated into their individual cells. Meiosis 2, we are then going to get our sister chromatids. And so we have our haploid cells N with our unreplicated chromosome. So we start with a diploid cell. We replicate the cells twice because we had double the DNA information, and then we replicate them again. We split them again. We don't replicate the DNA, but we split the cells again to end up with 4 sister chromatids that have a haploid amount of chromosomes, so 23. So again, meiosis 1, chromosome number reduced from 2N to N. This is all going through our different phases. We don't need to know this. But this, I think, is a better summary slide. So if you really want to remember, like, what is metaphase, what's telophase, I don't remember from genetics, I got you. This would be your meiosis 1, so your first step. So again, we have our pairs of chromosomes, they get replicated. So we have double the genetic information here. During prophase 1, we start getting them separated into the sides. We see our centromeres being formed. We see our spindle fibers being formed. In metaphase 1, that's when they actually get to line up in the middle, much like mitosis. Anaphase 1, we start seeing that pinching or that division, that pulling of the material from one side to another. And then telophase 1, we see pinching of the cell and separation. Mitosis 2 or meiosis 2 looks very similar. We have the same phases here. We have 2 separate cells undergoing these same phases. We have prophase again, metaphase, anaphase, and then telophase with cytokinesis. The key with meiosis is it's 2 cellular divisions, only 1 DNA replication. That's how this process works. And again, during meiosis, we also get increased genetic diversity because each daughter cell is genetically different from the original cell. When we do that replication piece, we allow crossover to occur, so it shares genetic material between chromosomes. So it makes a new genetic person, essentially, or new genetic zygote. And this is just comparison of mitosis to meiosis if you're really having trouble visualizing it. But again, the big piece of the puzzle here is meiosis has 2 phases. You're going to go through replication once, through cellular separation twice, and you end up with 4 daughter cells that are haploid versus 2 daughter cells that are diploid. If you're a video person, I do have videos of each of these phases, but we're not going to watch them for today. But if you are into the videos, there you go. That was your quick rundown of meiosis. Quick and dirty, got through it. Key piece of the puzzle, track your chromosomes. They like to do like haploid, diploid type questions. So that's a common one. Typically though, in association with some genetic congenital abnormality or disease. So now we're going to get into a little bit of the anatomy of the male reproductive system. So again, we have our testes, our sperm-producing male gonads that lie within our scrotum. And we have sperm is delivered to the body through different ductal systems. So understanding those ductal systems, they tend to ask questions about this. So epididymis, ductus deferens, ejaculatory duct, and urethra. And then there's the accessory sexual glands or sex glands, the seminal glands, prostate, and vulval urethral glands. So if you are a picture studier, again, we won't get a picture on the test, but sometimes it's helpful to have that visual association. You can see the different products and structures we're talking about here. So starting up top with the bladder, we have our ureter, peritoneums on top. This is our rectum, just to kind of give you perspective. Pubis bone right here. We can see these ducts coming down. So our ejaculatory duct here, our ductus deferens, vas deferens up front, urethra coming down this direction as well as this direction. Our epididymis here next to the testes and scrotum. And then on the penis, we do have different tissue layers here. We have the corpus cavernosum and the corpus spongiosum. Again, one of the pieces of the puzzle that I think is commonly asked is the corpus cavernosum is just on one top layer of the penis versus the corpus spongiosum is going to wrap around the urethra. It's going to be on more of the posterior layer, but it's going to be visualized on both sides of your urethra. So you can see here, corpus spongiosum tissue is up top, and then we have the addition of our corpus cavernosum tissue. So they often will ask the differentiation between those two on your NPlex exam. It's a common question. Glans, penis, distally, and then the prepuce or foreskin, and your external urethral orifice. Yes. Correct. Yes. So if you were asking any vascularity question, your answer would be cavernosum versus urethral type question, or where they often comes into play is like there is a laceration or a lesion or an injury to the tissue, this tissue of the penis. What structures could be affected, or what type of functional outcome could you see changed? And so if it was the corpus cavernosum, you'd be looking for things like necrosis or infarction or blood loss. If you're looking for things, if it was the spongiosum injured, you'd be looking at issues with urination or ejaculation. So then thinking about the scrotum and the testes and their functions, as well as some of their makeup. So there's two muscles that are involved, and we'll look at a picture that shows them, but the scrotum is affected by temperature changes. When cold, testes are pulled closer to the warm body wall. That's to protect our sperm production and protect all of those cells and the important actual molecules that then are gametes that then can lead to a zygote. That's why it gets pulled towards the body. So we have our dartos muscle and our cremaster muscles, and we'll see, I'll show a picture in the next slide, but actually I think I can't keep this here, so I'll keep my words and I'll go to our picture. So we can see here our cremaster muscle located here alongside the scrotum. Cremaster muscle is kind of these bands of skeletal muscle, and it will be involved in directly elevating testes. And we can see that here. And then the dartos muscle, okay, here's the layer of smooth muscle and superficial fascia that kind of wrinkles the skin around the testes. If you've done, I'll get to you. If we've done, I don't know if you guys have done urology yet and done like the male exam at all, DREs, male exam, hernia check, prostate exam, no hernia check exam yet. Haven't done repro yet. Okay, got it. So that the dartos muscle, kind of the fascia that allows for that wrinkling, makes more sense once you've done a hernia check, because you actually have to grab from that muscle, that like tissue, that fascia, to be able to do a hernia check without causing pain to your patient. What was your question? The only skeletal muscle that's involuntary or has an involuntary role. I don't want to say only, because I can't think of it at the top of my head, but it's definitely one of the major ones, I would say. It's a rare muscle in that it is involuntary, but there's also can be a not voluntary, but more of an involuntary response. If you do an exam where you actually stroke your finger up the inner thigh, you can see the cremaster, it'll be a reflex contraction that actually then moves the testes upward. And that's how you can tell if it's working appropriately. Yeah. But it would count as a skeletal muscle, but it almost have action more like a smooth muscle in that it doesn't, you're not voluntarily controlling it. Yeah. And then you have fibrous layers or tunic layers. That's what we call them. So the cremaster muscle, most important muscle to know when we're referring to testes. Then you have these two tunic layers, your tunic vaginalis, which is from peritoneum, and your tunica albuginea of the testes. And the tunica vaginalis, I think about it as the outside layer. It comes from the peritoneum, versus the tunica albuginea is the inner layer that forms more of that fibrous capsule. So it makes sense. If you can think about vaginalis being outside, external, and albuginea being internal, then you can recognize where it comes from and what it does, right? You just need to remember which one's external versus internal. There is this kind of septa of the scrotum that separates the two different sides from each other. And then you have your, where is it on this? Can we see this here? Your epididymis and your seminephros tubules. I don't know if it's on this picture. We'll go to the next one. So then when we're talking about the testes and really functionality, we're thinking about sperm production. So sperm is conveyed from our seminiferous tubules to the straight tubule, the ret testes, the afferent ductules, and the epididymis. So memorizing this process might be helpful if anatomy is your jam. There will probably be like female or male anatomy. More years than I can count, there's been more male anatomy than female anatomy on the NPLEX one, but that's not a guarantee it'll happen again. It just tends to be that way. That's what I keep hearing. And I can definitely attest to it was that way on my NPLEX one. The epididymis itself is made up of a head, body, and tail, and sperm are stored in the tail region until ejaculation. I appreciate this because the sperm also kind of have a head, body, and tail. So it makes sense to me that so does the epididymis. I think about sperm being stored in the tail because it's like where the movement is. And so that's where they hang out until they're able to actually be released. So blood supply specifically to testes. I'll show you a picture here in a minute that we'll kind of go through. But our testicular arteries do arise from the abdominal aorta. So that's their source. The testicular veins arise from the pampiniform venous plexus that surrounds each testicular artery. We love plexus, right? We love different venous collections in the body. And then we also have the spermatic cord, which encloses our nerve fibers, blood vessels, and lymphatics. So we can see this here. This is our spermatic cord, our different blood vessels and nerves. And then this is that process that we're talking about previously. So our ductus, our vas deferens. We can then follow it down here all the way through, and we can see tail of epididymis, duct of epididymis, body of epididymis. As we move up here, we're at the head of the epididymis. So you kind of scooch around. So this whole structure from here all the way down to here would be considered your epididymis. It's going to sperm will then move around here into the ductus or vas deferens, and then move up the spermatic cord during ejaculation. You could think about that, yeah. So when you're in your actual, you have seminiferous tubules actually within the testes. That's where your sperm is made here. So this seminiferous tubules is where the sperm is made inside your tunica albuginea. So you have your tunica albuginea, which is your inner fibrous capsule. Within that is the seminiferous tubules. That's where sperm production actually occurs. And then from the seminiferous tubules, they're moved through the straight tubule here, through the ret testes here, and into our epididymis, where they're stored. Essentially, they go through this kind of like region where there's the final production of sperm is made. We'll look at sperm production here in a minute. And then they move to the tail once they're fully formed, where they wait for that signal from ejaculation. And then they move up and out through the vas deferens. Typically, no peristaltic contractions are occurring. I mean, there is some smooth muscle that exists there that can kind of help move things along, but it's not, I wouldn't consider it peristalsis like the abdominal muscles, where it's like kind of this continuous, like, or the esophagus, where it's kind of this continuous rhythmic condition. It's more based off of concentration. So if there's a lot of sperm buildup, then you're going to see some contractions move the sperm down further towards the tail. But they almost move from the area of higher concentration to lower concentration based off of space availability. But that's why there's so much different little glands and ducts and spaces that they can essentially be moved down as they're made and formed until they're hanging in the tail waiting to be released. And again, you aren't going to have to identify things on anatomical structures, but if that helps you, this is a cadaver view. So a little aside on testicular cancer. It's a rare cancer, but it's most common cancer in men in aged 15 to 35. So if you're thinking about what's a younger younger cancer etiology that's more common in males, testicular cancer would be a good answer, because typically prostate cancer would be the most common cancer, right? So that we see in men in general, but it's occurring later on in life and it tends to not become metastatic or that severe. So if you get a younger patient that you're looking at, or if they ask you a epidemiology question for someone who is of younger age, testicular cancer might be a good answer for you. A risk factor is also a mumps infection that leads to orchitis, which is inflammation of the testes. They throw in an immune system question, which is always fun. And again, cryptorchidism or non-descent of the testes is the most common risk factor. So you do have this congenital or structural component too. And your clinical characteristic is a painless, solid mass in testes, typically can be cured due to surgical removal or radiation chemo. But this one's an important one just to keep in mind, because I think it's an easy question to try to get you to choose prostate cancer. Prostate cancer is probably the most common cancer. So again, we're going to get more into sperm production and sperm function here. So a reminder, our accessory ducts are going to carry our sperm from our testes. So once we got in that seminiferous tubule and we're waiting in the epididymis into the tail, then we're going to see release from the epididymis through the ductus or vas deferens. And I keep saying both because you see them written both ways on the implant. Yeah. And then moving through the ejaculatory duct and the urethra. So talking a little bit about where sperm are maturing. Sperm mature in the epididymis. They're made in the seminiferous tubules and they mature in the epididymis. The head of the epididymis is going to contain these different ductules that empty into this duct of the epididymis. There you saw it. It was very coiled, had lots of different ducts and areas for drainage. And then the body of tail is where typically they're stored. The majority of our ducts in the epididymis are made of pseudostratified epithelial cells. And they do contain some microvilli. So that's how you get some movement, is actually just that sweeping action. They also have larger surface areas, so you can get lots of different nutrients and blood flow and supplies to these sperm that are developing. The sperm are non-modal initially. That's why they can't move themselves down. Takes about 20 days when they're finally gain the ability to swim. That's why if you do any infertility or fertility counseling with patients and you do a sperm analysis, we do a calculation of modal versus non-modal sperm, or how modal are the sperm, how effective is their motility. It can tell us a few different things, but it can also say, are these sperm being released before they're fully formed and functional, or do they have a genetic inability to become modal, or is their motility just weakened? And so there's different things we can look at there. Some of those things we can overcome them if we're talking about fertility or infertility, and some we can't if it's really a genetic piece that we can't fix. But during ejaculation, that's when we do see the epididymis contract, and that's how we have expulsion of the sperm into our ductus or vas deferens. Our ductus deferens forms this ampulla, and it joins up with the seminal vesicle to form the ejaculatory duct. That's where smooth muscles in the walls really help propel, is during this process. And a vasectomy is cutting and ligating that ductus deferens. So that's what a vasectomy does. So again, visualization, I think, is the easiest sometimes for us. So we have our testes here, we have our spermatic cord here, our blood vessels and nerves. Within the testes, we have our tunica vaginalis, which is our outermost layer, our tunica albuginea, which is our innermost layer. We have our seminiferous tubules, where we have the formation of sperm. Sperm then is going to move through these tiny duct chambers, which is the straight tubule, into the ret testes, which is here. Then going to move into the head of the epididymis, where microvilli are going to help move it along, as well as just the normal natural flow of kind of these different ducts, downstream to the body, and then through these ducts to the tail of the epididymis, where sperm that mature and hang out for about 20 days till they can swim. They're then going to be released through our ductus deferens, our vas deferens, again through that spermatic cord. So the urethra is interesting in males because it conveys both urine and semen at different times, which is why it's an interesting structure. It has 3 different regions: the prostatic urethra, the intermediate part of the urethra or the membranous urethra, and the spongy urethra. So the prostatic urethra is surrounded by the prostate. That one's easy to remember. The membranous is in the urogenital diaphragm, which I'll show you a picture here in a minute. And our spongy urethra runs through the penis and it opens at the external urethral orifice. And the spongy urethra, the nice thing, it's in that corpus spongiosum. So if you can remember that the spongy urethra runs through the penis in the corpus spongiosum, that will help you with those tissue layers. So again, here we can see our prostate here. We can see our prostatic urethra running through. Then we have our urogenital diaphragm. That's kind of in this middle portion here, which is where you'll see that intermediate part or that membranous part of the urethra. And then the urethra is going to move down here through the penis, the body of the penis, and that's where it's in the corpus spongiosum. Yes. It could be. So it's part of the pelvic floor of males. So it is going to help if that you could potentially do male pelvic floor work, and that's actually becoming much more of a thing. People are becoming more comfortable with male pelvic floor work being done. Females, it's been done for a while. You use pelvic floor work to strengthen those different muscles, but it could help if it is more incontinence due to pressure. So pressure-based incontinence could be helpful there versus hormonal-based incontinence, or incontinence due to like an enlarged prostate wouldn't necessarily be effective strengthening that diaphragm. Yeah, absolutely. So then the penis itself, it's that main copulatory organ. We've seen it counts as part of the external genitalia, so scrotum and penis. And the penis, we've talked about the different structures already, so I'm not going to belabor this too much, but it does have that shaft that ends in the glans penis. And then there is the prepuce or foreskin, which is a cuff of loose skin that covers the glans. We've talked about the 2 types of tissue, but again, I'll mention corpus spongiosum surrounds that urethra, and it expands to form the glands in the bulb of the penis, versus the corpus cavernosa is paired kind of to the dorsal erectile body. So what does that look like? Again, so we have our spongy urethra here, and I'm seeing if we have a good picture. Our corpus spongiosum on either side, and then I'll change my color to make it easier. And then our corpus cavernosa right here and right here. And so if we look at this cut straight through, transverse cut, we can see corpus cavernosa here and here. I realize that blue is like kind of challenging to see. And then we can see here, this corpus spongiosum. So I think this view is a lot easier to just visualize, but externally it does not appear this way, right? But this view makes it a lot easier to visualize. Oh yeah, the spongiosum is directly around the urethra versus cavernosa, not so much. Cavernosa, and then those arteries that you mentioned here. We can see that in that cavernosa section. And I think that here, it's the same picture. We already covered those. I'm going to move forward. These are talking about the bones. I'm not going to spend that much time. Okay, great. So then accessory glands. This is what I wanted to hit now. So I want to get through our male accessory glands and get through a little bit of sperm formation before we break and move on to female anatomy. So our male accessory glands involve the seminal glands or seminal vesicles, which we talked about already, as they're on the posterior bladder surface. They contain smooth muscle that's going to contract during ejaculation. And they also produce this kind of viscous alkaline, so basic seminal fluid that contains a bunch of different stuff, including prostaglandins, some sugar, some coagulating enzymes. And again, the seminal gland joins the ductus efferens to form the ejaculatory duct. That's probably the most important thing to recognize about the seminal gland or vesicle. Our prostate encircles that urethra that's inferior to the bladder. That's why in benign prostatic hyperplasia or prostate cancer, you can see issues with incontinence, nocturia, urinary frequency, urgency, etc., because that prostate is going to It encircles the urethra, and it can also push on different structures if enlarged. It does have smooth muscle that contracts during ejaculation. It secretes our prostate-specific antigen, which is why we can measure that. It's going to help with sperm activation and makes about 1/3 of semen volume. That's not necessarily important for the NFLEX. And then our last gland is going to be our bulbourethral glands. These are pea-sized glands, very small, inferior, so below the prostate. They're going to produce more of a mucus, which is going to lubricate the gland's penis. It also is going to neutralize any acidic urine in the urethra. So this is helpful in preventing degradation of sperm, that maybe if there was an acidic environment, they might not survive. Yeah. Yeah, good question. The food for the sperm. So we can see here that this has stuff for sperm activation. This is going to be actually the food for the sperm because it contains our fructose. It's more alkaline. So you could think about this is the environment that sperm want to live in versus this is how they get activated. Because fructose, that's our sugar that sperm will use metabolically to survive as they go on their journey. Yeah, totally. Good questions. So we already mentioned this a little bit, but this is a common condition you might see. It's benign prostatic hyperplasia, prostatitis, or prostate cancer. So BPH, common disease, typically age-related. You'll typically see it in elderly men or men above the age of 50, but it can be younger. You see this benign growth of the stroma and glands of the prostate that can constrict the urethra, making, like I said, urination more difficult. Or incomplete emptying. A complication could be urinary tract infections or kidney damage. So upstream effects can happen. Prostatitis, this is a common reason for a man to consult a urologist. What I want you to recognize with prostatitis is it can be related to infection. It can be related to not infection. So it doesn't necessarily always have to be due to an infection. It could be chronic and inflammatory. And then prostate cancer is third most common cause of cancer death in males. It's a very common cause of cancer, though, in males. It doesn't kill a lot of males, but it's very common. And really, that's where that PSA level can be elevated. So just an aside on semen, just because we did talk about energy production for our sperm and then talked about the food for the sperm. So it does have fructose to allow for ATP productions for energy production. It's protecting and activates sperm, and it can help with movement. It is alkaline in nature, so if it's very acidic, that could potentially be harmful for sperm or less helpful. It does have these prostaglandins in there. It has the hormone relaxin. It contains ATP. It has some antibiotic chemicals, so it can have some antimicrobial effect. And there's also clotting factors that kind of coagulate semen initially and then liquefy it by fibrinolysin, so sperm can kind of finish the journey. So there's a lot of chemical makeup in semen. Don't memorize all of these things. Just recognize it has energy in fructose for ATP. It has some stuff that can help with antimicrobial resistance. It also is going to have things that help tissue relax and help sperm move easier. That would be the kind of the areas I want you to think about. Most common question I get is, can ATP production occur, energy production occur in semen? The answer is yes, because fructose is there, fructose is present. So it can, their ATP energy is present in that fluid. So we're going to talk a little bit about this. We already mentioned this in our autonomic nervous system discussion yesterday, but an erection is that enlargement and stiffening of the penis. Arterioles are normally the ones that are constricted. Sexual excitement can cause our central nervous system activation and then peripheral nervous system activation through a parasympathetic neuron, parasympathetic response, right? We remember erection is parasympathetic versus ejaculation sympathetic. So our pudendal arteries, that's what's innervated by our parasympathetic nerves from that sacral nerve root. It's going to cause local release of nitric oxide, which is a potent vasodilator. It's going to cause relaxation of local vascular smooth muscle, increased vascularity then to the penile tissue. So we have those arterioles dilate, then the cavernosa, given that that's the region where the arterioles are located, it's going to expand. It's going to lead to engorgement of the erectile tissue. And then the corpus spongiosum, its whole purpose is just keeping the urethra open. So then how does semen actually escape the system? So propulsion of semen through the male duct system, it's a spinal reflex. It's initiated from sympathetic nerves being generated. It's going to lead to the ductus deferens, the prostate, the seminal glands contracting and emptying their contents all into the prostatic urethra. The bladder and your internal sphincter muscles also going to constrict, preventing urination, right? So that's how it's a unilateral system. That's why we don't urinate and ejaculate at the same time, which is good. The semen in the urethra triggers that spinal reflex through a somatic neuron. Remember, somatic is muscular contraction versus autonomic. Then our bulbospongiosus muscles undergo kind of this rapid contraction series that allows for expulsion. Because it's a sympathetic response, that's why you can see increased heart rate, increased breathing, etc. Afterwards, there's a period of resolution where muscles relax, arterioles and arteries constrict. And then there's this latent refractory period where the process is unable to occur again right away. So in ED, erectile dysfunction, there's many causes. So this is just one, but parasympathetic nerves of the penis are releasing too little nitric oxide, leading to the inability of attaining an erection. That's why Viagra was created, to allow for a vasodilation to the tissues and increased nitric oxide production locally. But it's also not the only cause of ED. So some men it doesn't work. So again, certain meds or other substances can contribute. Any condition that affects blood vascularity, so atherosclerosis, hyperlipidemia, hypertension, diabetes, can cause or contribute to ED. Even nerve damage, multiple sclerosis. So there's many different causes that contribute, but typically when they talk about them in relationship to different heart or cardiovascular conditions, because it's easy to ask questions that kind of cover both areas. Or diabetes. I feel like when in doubt, diabetes is asked with everything these days. Yeah. It doesn't, that would not be related to nitric oxide release because we're not necessarily affecting that region. If you're actually removing the prostate, you're removing some of the area of production of sperm and semen. You're also removing some of those control factors that could stimulate a erection. You're also going to be decreasing localized potentially testosterone and hormones. So there's kind of multitude of factors. And then the physical scar tissue piece too. So I'd say that would be multifactorial cause of ED. It can, absolutely. But I wouldn't hang your hat on that being one etiology. So that would be a multifactorial component. Yeah, good. All right, and then before we break, I want to just go through a little bit about spermatogenesis. We talked about where sperm are formed, but I do want to spend a minute on talking about some of the processes of formation of sperm. So spermatogenesis is our process of forming our male gametes. Like I mentioned, it occurs in seminiferous tubules. It starts around puberty. So what do the seminiferous tubules look like from a histologic sense? Yikes, right? Histology, but they love asking about cell types. We've seen that already, right? So let's just think about what the tubules contain. They consist of this thick stratified epithelium, surrounds a central fluid-filled lumen, and it has 4 different important types of cells that you should kind of have in your head. Stenocytes, spermatogenic cells, myoid cells, and interstitial endocrine cells, or Leydig cells. And they'll probably call them Leydig, but they could call them interstitial endocrine. So we'll go through each. So stenocytes are columnar cells. They're large, they're supporting cells, and they play a role in sperm formation. That's really all I want you to know. They're large and they're kind of the supporting actors. Spermatogenic cells are surrounded by cystinocytes, but spermatogenous cells are the main cells that give rise to sperm. So you can think about spermatogenic cells being the ones that are really creating the sperm, and the cystinocytes are standing next to them, supporting them, holding them up. Your myoid cells are muscle cells. They're going to surround that seminiferous tubule, and they're going to actually help squeeze sperm and fluid through the tubules. Myoid, I think muscle starts with an M, so that's helpful. And then your interstitial or Leydig cells are going to be those endocrine type cells. So they are going to produce your androgens and a little estrogen to help control this process, increase or decrease. So if you wanted to see visually what that looks like. So within the seminiferous tubules, so this is where we are. We took a tiny little picture, and we can see our spermatogenic cells here, these little dark purple cells here. We can see our cystinocytes. They're not really necessarily super clear, but it's kind of this lighter pink portion surrounding or next to our spermatogenic cells. We have our interstitial endocrine cells on the side. They almost have like some space within them. And then our myoid or muscle cells are thinner. That's just one seminiferous tubule. And this is just a different view. So in the seminiferous tubules of the testes, our spermatogenic cells are going to give rise to our sperm. So there's three major steps of spermatogenesis: mitosis of spermatogonia, which is a stem cell that forms two spermatocytes; meiosis itself, which is where the spermatocytes form our secondary spermatocytes, which form spermatids; and then spermatogenesis, where spermatids become sperm. So these are our three major steps: mitosis, meiosis, and spermiogenesis. Okay. So mitosis is essentially of our spermatogonia. From mitosis, we get spermatocytes. So if you remember one thing, remember that. Spermatogenesis begins at puberty. You start with spermatogonia, which are stem cells. They're going to divide more or less continuously by mitosis. So our spermatogonia, our stem cells, and they divide by mitosis. After puberty, each division is going to give you as a type A or a type B daughter cell. Type A daughter cells remain at that basal lamina, and they continue to do mitosis. They continue to divide. Type B daughter cells move towards that lumen, and they're going to develop into your spermatocytes. So in mitosis of spermatogonia, step one of spermatogenesis, you're going to have spermatogonia, which are stem cells dividing like mitosis. After puberty, you get type A and type B daughter cells. Type A daughter cells continue to do what the spermatogonia are doing. They continue to divide using mitosis. Type B daughter cells move away towards the lumen, and they develop into primary spermatocytes. So this is the process visually, the whole process here. We just talked about our spermatogonium and our mitosis and our type B daughter cell. So this is what we've talked about so far. Step two then is our meiosis, where our spermatocytes become spermatids. We have obviously meiosis 1 and meiosis 2. So our primary spermatocyte that's diploid undergoes meiosis 1, forming two secondary spermatocytes that are haploid. So we have to form gametes here, right? That's our goal at the end of this. Then we have meiosis 2, where our secondary spermatocyte does meiosis 2 and becomes two spermatids, which are also haploid. Spermatids, they're small little round cells. They have a large nuclei. They're found close to the lumen of the tubule. We can see type B daughter cell, primary spermatocyte, secondary spermatocyte, spermatid. These are early spermatid versus late spermatids, almost really look like fully developed sperm. So our next step in spermiogenesis, that's when our spermatids become fully developed sperm. Our spermatids are still haploid, which is the correct chromosome number needed for fertilization. They're all non-modal at this point when they're spermatids. Through spermiogenesis, they elongate, they lose excess cytoplasm, and they form a tail. So they really have that, become that process where they have that head that's pointed, that body, and that tail. And that's from losing excess cytoplasm. So they become from the circle to an actual fully developed sperm. The head has the genetic region and this helmet acrosome, which has these hydrolytic enzymes that allows a sperm to penetrate an egg. The mid piece is their metabolic region that can produce ATP using typically fructose. And the tail is the locomotor region that contains flagellum. So head with acrosome contains genetic info. Body or mid piece is genetically or is metabolically active. And then your tail is your mobility center. It's going to take about 64 to 72 days if conditions are hospitable, meaning if they are ideal conditions, and sperm are unable to swim initially, but pressure of the testicular fluid pushes immotile sperm into the epididymis where they gain that motility and fertilizing power. So in the side on our sustainocytes are also called Sertoli cells. That's where our Sertoli cells come into play. So our Leydigs, remember, are endocrine cells that help with our testosterone and estrogen production. Our sustainocytes are our Sertoli cells, our big supporting cells that surround our developed spermatogonium. They have two separate compartments. Really what I want you to know with sustainocytes is they help kind of form this blood-testes barrier. They prevent any antigens from sperm from getting into the bloodstream and causing an autoimmune response to self. And since sperm is not formed into puberty, it's absent during immune system development. So if these Sertoli cells did not exist and sperm components or antigens did get into blood flow, we would not recognize them as self and we would have an autoimmune response. Sustainocytes also, because they're large, they're columnar, they can provide nutrients to dividing cells. They can help with movement. They can secrete some testicular fluid that has androgens and some acids into the lumen. They can phagocytize germ cells and excessive cytoplasm. And they produce these two mediators to regulate androgen binding protein, which keeps testosterone levels high, and inhibin, which we've talked about already. So our Sertoli cells, they do pretty much everything. There's almost nothing Sertoli cells don't do, but except for actually produce sperm, right? They're the supporter cells in every way, but they themselves are not producing sperm or spermatogonia. I'm moving through infertility. I'm seeing if there's any other points I really want to hit before we're done. Oh yeah, regulation, and then we'll take a break. So we already talked a little bit about the HPG axis. And so this process here, we see hypothalamus is going to release GnRH. GnRH releases FSH and LH. They are going to indirectly stimulate spermatogenesis by causing sustainocytes to release that androgen binding protein, which keeps local concentrations of testosterone high. LH then can stimulate interstitial endocrine cells to secrete more testosterone, which is needed for spermatogenesis. So we have two major ways of getting testosterone happening locally. Testosterone is then going to act on other body sites to stimulate maturation of sex organs, development, maintenance of secondary sex characteristics, and libido. There is some negative feedback by testosterone, which inhibits FSH and LH, and also GnRH release. And then inhibin release locally by sustainocytes also is going to feed back onto the anterior pituitary, decreasing FSH release. So you have localized and you have systemic negative feedback control. If you're not a visualized person and you want to read it, this is kind of talking about the same thing just in words. And then this shows kind of your overview of what these hormones are doing testosterone level wise, specifically through stages of life. So at fertilization, if they're low, it's going to bump up. And then birth, it's low. You have this little spike, and then it goes down, and then it's going to stay low, low, low until you hit puberty, and it flies up nice and high until adulthood, and then starts to decrease over time as we age. So testosterone does come from cholesterol. It's converted to dihydrotestosterone, DHT, in the prostate, and estradiol in some brain neurons. That's why men have estrogen too. Again, it prompts spermatogenesis and affects our target organs. It has had anabolic effects in the body. That's why people can abuse testosterone, right? Inject it. And deficiency leads to atrophy, decrease in semen volume, decrease in or effects on erection and ejaculation, which is why testosterone repletion has existed. And since we will talk about female secondary sex characteristics, it's important to understand males have them too. Mainly testosterone is contributing to these. And so this is your different hair, deepening of voice, thickening of skin and oily skin, bones growing, increasing in density, skeletal muscles increase in size and mass, and increase in BMR. Did it. Male stuff, male anatomy. We made it through. Highlights, understand those different tissue structures we leaned into. Understand spermatogenesis. If you have those two parts down, you'll be pretty well set up for male reproductive system. All right, we're gonna take a 7-minute break. We'll come back at 9:11 and we'll move into female anatomy, or female reproductive system, which is the next PowerPoint 8B. I'll see you at 9:12.